The structure and function of the cell surface membrane. In this video, we're going to be talking about the many different functions of the cell surface membrane and then how it is structured to enable it to carry out those functions. Let's start with looking at the functions. Number 1. It acts as a selectively permeable membrane, controlling the entry and exit of substances into and out of the cell. It allows small, non-polar molecules to travel readily across the phospholipid bilayer, and it contains transport proteins to allow larger or water-soluble molecules to travel across. This can either be by facilitated diffusion or by active transport. You'll learn a little bit more about facilitated diffusion and active transport later on. One of the things to remember, and one of the common errors that students make when talking about transport proteins, is they sometimes say that they have the same shaped binding site as the molecule that they're transporting, but it's really important that you remember that they have a complementary shaped binding site to the molecule that they're transporting, and that's a really important term, complementary shape. Let's just give a couple of examples of each of these types. So when we're talking about small non-polar molecules, we could be talking about, for instance, oxygen molecules. We could be talking about carbon dioxide molecules. We could be talking about fatty acids and glycerol. When we're talking about larger or water-soluble molecules, we could be talking about things like amino acids, glucose, other types of simple carbohydrates like sucrose or fructose. Number 2. Taking up nutrients. This is intrinsically linked to the first point. And when we're talking about taking up nutrients, we could be referring to things like amino acids, fatty acids, glycerol, simple sugars like glucose and fructose, and we could also be referring to ions like calcium ions, sodium ions, potassium ions, and a range of other ions that the cell needs in order to function correctly. Number 3, secreting chemicals. For instance, a cell might secrete an enzyme. Enzymes that are secreted out of cells are known as extracellular enzymes outside of the cell. For instance, enzymes of our digestive system, like pepsin and trypsin, these are extracellular digestive enzymes that are secreted by cells. Some cells in your body are important for secreting hormones. For instance, insulin is secreted from cells in your pancreas. Estrogen is secreted from cells in your ovaries. So hormones are secreted from cells into the bloodstream where they are transported to their target organ where they will perform their function. Another chemical that might be secreted from a cell is a neurotransmitter. Neurotransmitters are the chemicals that are released from neurons. They're released into the synapse, and their job is to then transmit a chemical message from one neuron to the next, and they're really, really important in nerve transmission. Plasma B cells that are part of your immune system produce and secrete Y-shaped antibodies. And as you already know from GCSE, antibodies are small proteins whose job it is to bind onto antigens which are on the surface of pathogens. So when we talk about that term secretion, we are secreting, we are releasing useful substances from cells which are going to be used elsewhere in the body to perform a particular function. Number 4, cell recognition. The cell surface membrane acts as a binding site for antigens. Antigens are specifically shaped molecules, usually proteins, which can be recognized by the cells of your immune system to enable your immune system to determine self from non-self cells and therefore to protect you from invading pathogens. Number 5, cell-to-cell adhesion. In this context, adhesion means cells sticking together to form tissues. So in most of the tissues in your body, bar perhaps your bloodstream, we need our cells to stick together in order to perform their function. For instance, epithelial tissue. All of your cells stick together in the epithelial lining of your body, and that's really important. You need all of your muscle cells to stick together in order to form muscle tissue. You need all of your skin cells to stick together in order to perform their function as skin. Number 6, hormone binding. Protein-based hormones, for instance, insulin, oxytocin, and leptin, bind to receptors on the cell surface membrane. These receptors have complementary shapes to the hormones to allow them to bind on. These hormones will then trigger a cascade of chemical reactions within the cell to enable them to perform their function. Lipid-based hormones can just travel directly across the phospholipid bilayer, and they don't need to bind onto a receptor on the surface. I've obviously given you the names of these hormones, but don't worry about those. You don't need to know those names specifically. I've just given them to you out of interest, and if it's something that interests you, go ahead and research them separately. Okay, we're just going to take a little sidebar for a minute now and just talk about the functions of membranes within cells. And when we talk about the functions of membranes within cells, we're really referring to the membranes surrounding the organelles within the cell. So almost all of these functions are almost identical to the functions of the cell surface membrane. There are just a few additions, so we're going to touch on those. Number 2, separate organelles from the cytoplasm so that the reactants for specific metabolic reactions are isolated. We don't want the enzymes involved in one chemical reaction that might be occurring in the chloroplast to get in the way of or disrupt a metabolic reaction that's occurring in the mitochondria or in the lysosome or in the rough endoplasmic reticulum. So it's really important that these chemicals are isolated to prevent metabolic reactions being disrupted. Number 3, provide internal transport systems. For instance, the rough endoplasmic reticulum and the Golgi body provide a transport system for proteins as they are being translated and being modified so they can be used within the cell or secreted out of the cell. Number 4, they can isolate enzymes that might otherwise damage the cell's contents. This is done within lysosomes. For instance, lysosomes contain hydrolytic enzymes like proteases and lipases that would otherwise digest the contents of the cell. Number 5, they can provide surfaces on which reactions can occur. For instance, the rough endoplasmic reticulum provides a surface on which protein synthesis can occur. The inner mitochondrial membrane provides a surface on which the electron transport chain can occur, which is the final stage of respiration, something you don't need to worry about until year 13. The inner membranes, the thylakoid membranes of the chloroplast, they provide a surface on which the light-dependent reaction of photosynthesis can occur. And again, you don't need to worry about that in too much detail until year 13. Let's move on to talk about the structure of the cell membrane. The current model of the cell membrane is known as the fluid mosaic model, and this teaches us two things about the membrane. One, fluid. It teaches us that it is melted at body temperature. And two, it is a mosaic. That means that there are molecules embedded throughout the membrane. And because the membrane is fluid, these molecules have freedom of movement within the membrane. It tells us that the membrane is flexible, and it tells us that the membrane is self-sealing. Things can be added to the membrane and things can be taken away from the membrane. And this is a really important concept to understand as you go on in biology and you learn more and more about the processes that occur within and between cells. If we look at the membrane on the most basic level, it is made up of a phospholipid bilayer, and so we must first look at the structure of the phospholipid. A phospholipid is made up of a phosphate head attached to a glycerol molecule and two fatty acids making up the tail. Each one is about 3.5 nanometers long, so that tells us that the phospholipid bilayer is about 7 nanometers in diameter. The phosphate head is hydrophilic. Hydro meaning water and philic meaning loving. It is a water-loving molecule, and it will always face outwards because the cell membrane sits in an aqueous solution. The fatty acid tails are hydrophobic, water-hating. They will always face the inside of the membrane. If we were to be able to take a handful of phospholipid molecules and just drop them into a bowl of water, they would spontaneously form into a sphere of a phospholipid bilayer with all the fatty acid tails on the inside and all the heads on the outside, purely due to those characteristics of the molecules that make it up, the hydrophilic head and the hydrophobic tail. Just looking at this really basic diagram of the phospholipid bilayer that I've drawn here, it's really important that you understand that you can only see this level of detail under the electron microscope. This is because the light microscope does not have a high enough resolution to enable you to be able to see this level of detail. One little extra note about when you look at the phospholipid bilayer under the electron microscope, what it will look like is look like you're seeing two lines, two dark lines with the hole in the middle. This is because when you look at it under the electron microscope, you're seeing the phosphate heads but not the fatty acid tails. This is because the stain that scientists use to enable them to see the cell membrane binds to the phosphate heads, but it doesn't bind to the fatty acid tails. Before we move on, let's just talk a little bit more about the properties of the phosphate head and the fatty acid tail. The phosphate head is a polar molecule, whereas the fatty acid tail is non-polar. It is neutral, and therefore this molecule is amphipathic in nature. It's really important that you understand that the only things that can move directly across the phospholipid bilayer without the aid of any transport proteins are fat-soluble or water-insoluble molecules. Let's now look at the structure of the cell membrane in more detail. I'm going to draw out a diagram of the cell membrane and then we're going to talk about it together. It's really important when you look at the diagrams of the cell membrane that you try in your head to imagine this as a three-dimensional object, because of course on paper we can only draw it as a two-dimensional object. Let's talk about the functions of each of these structures within the membrane in turn. Glycoproteins and glycolipids are involved in stabilizing the membrane structure. So they form hydrogen bonds with water molecules surrounding the cell, and by doing that they stabilize the membrane. They're also involved in cell-to-cell adhesion, i.e. allowing two cells to stick together, which is really important for tissue formation. And specifically, glycoproteins can act as antigens, which are really important in cell recognition, and they can also act as receptor molecules, so as binding sites for other chemicals like, for instance, hormones or neurotransmitters. Cholesterol is a lipid molecule within the membrane. Cholesterol is involved in controlling membrane fluidity. The more cholesterol present, the less fluid the membrane. Linked to this, it controls the permeability of the membrane. The more cholesterol present, the less permeable the membrane. Cholesterol molecules are hydrophobic and therefore they prevent leakage of water or dissolved ions from the cell. They prevent the movement of water and dissolved ions into or out of the cell. Linked to this, they are involved in membrane stability. So more cholesterol equals a greater membrane stability. With no cholesterol at all, the membrane would be totally unstable and the cell would just burst open. So cholesterol is a really important molecule, but depending on the function of the cell, there will be different concentrations of cholesterol. Let's look at the phospholipid. We know already that the phospholipids act as a barrier to most substances and therefore help to control what enters and exits the cell. The phospholipids also make the membranes flexible and self-sealing. This is because of their amphipathic nature, the fact that they have hydrophilic heads and hydrophobic tails. So let's look at what can and cannot travel across the membrane. Small non-polar molecules can rapidly diffuse across the membrane, things like oxygen and carbon dioxide. Small polar molecules, such as water and urea, can diffuse across, but much, much more slowly. Charged particles, ions, or water-soluble substances cannot diffuse across by simple diffusion. They either rely on facilitated diffusion, so the presence of transport proteins within the membrane, or active transport, which also requires transport proteins within the membrane. And that's essentially everything you need to know at this point about the cell membrane. Well done.